I'd like to begin by acknowledging the traditional owners of the land on which we meet today. I'd like to pay my respects to the elders past and present, and I'm hosting this event from Irukandji territory. So welcome to our National Science Week 2020 event. This is fresh from the field, bat colonies, and live from the lab poo analysis. This Inspiring Australia event is supported by the Australian Government and it's hosted by the Australian Society for Parasitology. Now, my name is Lisa, and joining me today is Associate Professor Michelle Power from Macquarie University. Now, Michelle is going to be talking to us about poo this morning. Welcome, Michelle.
Hi, Lisa. Thank you. And before I move along, I'd also like to acknowledge that I'm here in Sydney and I'm on the lands of the Watamuggal clan of the Darug Nation. So thank you, Lisa. All right, so welcome to our lab here in Biological Sciences at Macquarie University. We do a lot of work in this lab. My research and that of the people who I work with is around understanding host-parasite interactions in Australian wildlife. So I also get to go out and do a lot of fun things in the field, and you'll see some of that in a moment. All right, so but before we trek on to getting into the field, we're actually going to have a bit of a chat about what is a parasite. So a parasite is an organism that lives in or on another organism. So for example, we've got this little parasite here, it's called Eimeria, and it lives inside the stomachs of kangaroos. Now, parasites are extremely diverse. There's over 80,000 different species of them, and they infect a whole range of organisms, not only wildlife, but plants, invertebrates, all sorts of other animals, and also us.
What animal's that you've got in the background there, Michelle?
That animal there in the background, Lisa, is an eastern grey kangaroo. It's a baby one.
And what parasite is that below it?
The parasite is Eimeria, and Eimeria is a protozoan parasite. It's a single-celled parasite that, as I said, it lives in the gut and it causes diarrhea.
In kangaroos or people or both?
This one isn't found in people, but it's found in a lot of different wildlife, including kangaroos, possums,
Okay.
Around the world, chickens, not just marsupials, sorts, and not just wildlife, all sorts of different hosts, chickens, sheep, cows. Yeah, it's a very ubiquitous parasite, this one. And
so not good for the animals, obviously.
Yes, if they have too many of them, it can impact their health. The interesting thing about this parasite is that it's co-evolved, or it has occurred with its host for a very long time. And so in some cases, it doesn't necessarily always cause harm to the host, which is the definition of a parasite. It causes harm to the host, but the parasite benefits in some way. So it gets a home, it gets resources or its food or dinner from living in or on that host.
Right.
Cool. But working on parasites means we actually have to get the parasite material. And when you work on gut parasites like I do, it means we need wait for it, their poo, okay? So I've used some tame photos here, and just behind me, my shoulder there, we've got a possum poo that's sitting on in a park. It's actually in Victoria, this one, and this is one of the sites I get to go to in the field. I know it doesn't sound that exciting, it's in the city, but we'll talk about some of the more interesting parts of my work relative to field work in a moment. This one up here, and if you can just make it out in the middle, that's actually a penguin poo. And that photo was taken in Antarctica, of all places. So that's one of the exciting places I get to go. So we go out into the field and we need to basically collect the poop of the animals that we're working on. So I have been, in my career, worked on lots of different wildlife and gone to lots of different exciting places, not just for field work, but also for conferences and for research collaboration. So being a scientist means that you actually usually get to travel quite a lot.
Not quite so much at the moment.
Not quite so much at the moment. So making the most of being at home or in New South Wales at the moment.
Oh, can I just ask a question? When you're going and collecting poo, if you haven't seen the animal actually doing the poo, how do you know that that poo belongs to, say, a penguin and not another animal that's living in the same area?
Yeah, excellent question there, Lisa. And you know, there's actually books on that, and there's guides, just like there's guides to
guides.
Well, yeah, just like there's guides to wildlife. There's I've actually got them in my office. I could have brought them in. There's a book called Tracks and Scats, and that's a guide to footprints of Australian animals and also the scats that they leave behind. And even some local areas and national parks also now have little fold-out scat guides.
What poo is that?
Yes, what poo is that? Exactly. And how do you know? I should have had more on display for everyone to see, but sometimes it can gross people out. Poo is actually, from wildlife, is often an indicator of where it came, and we use its characteristics, so its shape, its size, what it looks like. So possums do very characteristic poop, and theirs is it's quite elongate, it's about 20 centimetres long, whereas kangaroos are a little bit more cuboid, and wombats are the best actually because their poo is actually square, and they actually always position it in a place to set up a barrier and their defence areas and let people know that this is where they are. So you often see wombat poo up on a log. So basically the size and shape tells us what animal it came from. But we can also do DNA tests as well.
20 centimetre poo?
No. The size of a 20-cent piece.
I'm thinking, that's a huge possum poo.
Did I say it was 20 centimetres long?
No, just my ears. I'm going deaf.
I said it's elongate and it's the size of about the size of a 20-cent piece. Yeah, it would have been maybe a dinosaur.
I'm glad I clarified that.
Megafauna. We did have megafauna in Australia many years ago, so that's possible. 20-metre poo. That's a lot of parasites. All right. So, and the other thing for you people out there who have joined us today is parasitology is a really fun field to work in Australia. Okay, so these are Australian sea lions. Back to the field. These are Australian sea lions. So we've got a project happening in working with collaborators at the University of Sydney. So science is often about working with different people, different strengths, different areas. And I work with the vet over there, Rachel Gray. And this is the exciting one. This is some photos from Antarctica and trying to understand health and disease in Antarctic wildlife. So I got to go to Antarctica. But the work we're really doing at the moment is actually looking at parasites in flying foxes. And flying foxes are one of my favourite species. They get a lot of negativity around the risks of the diseases that they carry to people. And also because, we've given them a great home by building cities. And unfortunately, flying foxes are moving into cities because we're destroying their homes that when we clear land that they're used to. So we're trying to understand, so a lot of conflict and a lot of misconceptions around the risks that flying foxes do pose to us. I'm not saying they don't carry diseases, but, if we can manage and understand what's happening, we can control those risks. So we get to go out in the field and we do catch flying foxes and take samples from them, and then we let them go again. They're vulnerable and threatened species, and so we're trying to understand their health for the health of them and their conservation. So this is a photo taken down at one of our field sites in Adelaide, so I get to go interstate. And here's a fantastic photo actually taken by a collaborator who works up in Cairns, where some of our listeners may be. All right, so I'm going to stop talking and we're going to actually go into the field, and we're going to...
This is a live that you recorded previously, isn't it?
Yeah, so this is something, we're not live now. It's a pre-recording of some field work that we did and how we operate in the field.
Yep.
So we'll play this and just give me a moment to get it all together. Hopefully you can see that now.
Perfect.
Oh, and I need to press play, don't I? There we go. Here in the field, looking for bat poo. First of all, before we look for the bat poo, we might actually look for bats. Now, usually you might be used to bats being in your backyard and coming in late at night or seeing them fly over your houses. But where do they go in the day? Up in the trees. And we also know where they actually live in the day. So we can come to their colonies and we can actually set up the night before they fly out to collect nice fresh poo in the morning. Okay, so we've returned to the bat colony the next morning. Our tarps have been down overnight and the bats have also come back. They're roosting in the trees above us. Before they go to sleep for the day, they will poo and it'll be collected on the tarp.
Where was that footage taken, Michelle?
That footage was taken in a colony in southern Sydney.
Right. Okay. We've got a listener who was commenting on it. She thought it was maybe in Cairns, and I was thinking,
no, it's not in Cairns. So we're down in Sydney doing the field work down here. Obviously, we weren't doing that field work at that point in time, but it's one of the sites just in one of the parks around Sydney, and we have done we've worked and collected samples in Cairns as well, and worked with the Tolga Bat Hospital out that way within our research. Yeah. So yes, and I wasn't in Cairns. I know it's all palmy, but no.
Yeah, no, there was a tree there. Was it a camellia? And I thought, well, that won't be Cairns, but But when your plastic mat that you had down, so I assume you reuse that again for another. So you have to obviously clean it before you use it.
Actually, no, we don't. So we would set so we would go into the colony and they're usually across a wide span of the trees. We would lay down about 8 to 10 of those tarps and and then in the morning there will be just poo everywhere because there can be thousands and thousands. So we could go through a process where we wash it all off and bleach it, but we do need to also consider work health and safety and the risks of working with bat poo for us as well. And cross-contamination for our research. If there's a little bit speckle left behind, we might get skewed data. So no, we actually do dispose of those. So,
Michelle, we do have a question. I don't know that you'll be able to answer it, but because I'm not sure if you work on this area, but in terms of the gut biome of the flying foxes, is there a difference between metropolitan colonies and wild colonies, as in out in the bush, I suppose?
Yeah, that's actually a fantastic question. And at this point in time, no, that's something we are doing. We're actually, we're looking at the gut microbiome of flying foxes. So if people aren't quite sure, other listeners and people who have joined us aren't quite sure what that means, it's basically we've got a whole community of bacteria living inside our stomachs, and they do important roles around digestion and protecting us. They prime our immune system and so forth. And they also protect from or may change, that community may change with infection from other organisms, or they also can protect from other infections. And so we're trying to understand the interaction between the gut microbiome and parasites, not just in bats, in other species that we're studying as well. But coming back, I can't answer that to bats, but there is definitely some in other species, and I think polar bears outside of Australia, where people have actually shown that wildlife microbiomes do vary if they're grazing in city areas compared to in their habitats that haven't been impacted by humans.
Wow,
So,
that's amazing.
yeah, it's pretty cool. Yeah, and you know we're getting a lot of new technologies and that allow us to really look using DNA, like basically not looking at the organisms at all, look at using DNA and technologies to understand those communities and interaction. And I should say, folks, we are live from the lab. I am standing in here, so and we're going to see some action in a minute as well. So if there's a bit of background noise, I do apologise, but everyone's working away, social distancing, as a note if you hear bangs and things going on behind me. All right, so are there any more questions, Lisa, before we move on to into the lab?
No,
Okay.
we've just covered those. Thank you, Michelle.
Okay, alrighty. So what we're going to do, I'm just going to come back. Okay, so we're back live in the lab. Okay, we'll take away my virtual backgrounds. And so I'm going to attempt, this is a first run, I'm going to give you a tour, a pre-recorded tour of the lab, and I'll talk through some of the instruments that we have here and what we do once that poop sample that we collected or that you saw me collect, once that comes into the lab, what we do. And then we'll cross to one of the Amara, who's working next to me. We might not see her, but we'll definitely see her hands. I'm not sure where the camera's positioned. And she will be demonstrating some of the steps of a DNA extraction. So I'll just share another video. Oh. Sorry. Here in the field, looking for, hang on, technical, I've got multiple screens open here. Okay, the lab tour, here we go. All right, so you can see the sign that is behind me, and so it's moved. Okay, so in the lab we do look at parasites, and when we're looking at parasites at their form, we use microscopy. So here we've got a tapeworm. I actually think this one's from an Australian sea lion. And
wow.
Yep.
What is it?
Ian Beveridge.
It looks like pasta.
It looks like pasta. It does look like pasta. It does indeed. It looks like fettuccine. So we need microscopes to look at macroparasites, so worms, but also when we're looking at those protozoa that I mentioned, like Eimeria. So we've got some high-powered microscopes to do that. These are the sample tubes, one of the sample tubes that they come into the lab. But when we're looking at the organisms that we're trying to understand, we actually need their DNA. So we have to extract their DNA from the fecal samples when we come back in the lab. We do this using a kit and basically take the sample, pop it into one of these tubes, and then go through the DNA extraction process. Now that's what we're going to come back at the end live, which is Amara is doing right next to me. So, but this is this piece of equipment is a biosafety hood. We use this to protect ourselves when we're working with the organisms because because there's the potential risk that there is some organisms in there that may cause us harm. One of the other components that's really important to our work is the micropipettes that you can see here. And this is the setup for the DNA extraction. Just so you know, I had to get a circle in the lab basically. So we're just turning around, but you'll see what's happening now. This was the setup about half an hour ago. And Amara is working behind me. This is a long shot of the lab. And I actually have some people standing out to me at the right there. Back to the microscope. We've got balances. We need to make up chemicals. Sometimes we need to weigh poo. This is a very important piece of equipment, the centrifuge. And for the DNA extraction, we go through a series. We do it in small tubes. And we go through a series of steps where we actually mix the poop sample with buffers, apply it to a column, and then eventually our DNA sticks to a membrane, and then we do washes. And basically the centrifuge passes liquid over a membrane and is important part of that step. More microscopes, chemistry areas in the lab, special hoods for working in sterile environments, all our fridges and freezers, okay. Our labs are quite big here, as you can see. And then coming along to some other important equipment, more centrifuges, heating blocks, more balancers. And then, as I mentioned, we're extracting the DNA, but our final step to actually do the test for the parasites involves a process where we make hundreds of thousands of copies of a small part of the DNA that actually is a barcode or a tag for the parasite that we're interested in. And we do this using a process called polymerase chain reaction, or PCR. This is the machine. We can run 100 samples at a time. And we've got, you can see here, we've got 3 of them, but this is an important piece of equipment, and well, literally all labs would have one of these machines now, I would expect these days. And what else have we got? Right, so I think this is kind of closing towards the end of the lab tour and the other side. And so you're about to come in here. And so this is here. And if we flick over to the GoPro, you should now be able to see Amara, who is live and doing a DNA extraction. Any questions there, Lisa?
Yeah, you just need to stop sharing your screen.
Yep,
And I've got one.
Oh, sorry. Yeah.
And I've got one question. I think it's actually related to the previous one about the microbiome of the bats. Does it make a difference, the fact that the bats are migratory, as opposed to, you know, the polar bears? Like, do they live in that metropolitan area, or they, you know, do the bats who live out in the wild, but and also come into the metropolitan area, then go back out to the wild? Would that, do you think, make a difference to their microbiome?
Yeah, so I'll leave the camera on Amara and just answer this verbally, like without seeing me. So look, I'm not aware of any, so understanding or asking questions like that brings us to the scientific process, where we literally go out, collect samples, bring samples back to the lab and do rigorous testing to really address questions and gather scientific evidence to find answers, okay? I'm not aware of studies that have been done comparing the microbiomes of bats that are residents in cities versus those that are migratory. Look, it's a fantastic question. It would require quite a large amount of funding, because doing those microbiome tests, it's about $100 a sample, and that's just to get the microbiome sequencing. So look, I agree, it's a fantastic question. Given that the microbiome is driven by the host and the types of plants that it eats, it's potential that the diversity of the microbiome in bats fluctuates anyway over time relative to the plants that they're eating. So it's actually a very hard question to get to, because you've got the complexity of their diet, the complexity of where they're living at certain times, and then the complexity of interactions with what's happening in the world around them to actually get to that. So, but I think it's a great scientific question. But yeah.
Okay, tell us what is going on in the lab.
Yeah, okay, so Amara is basically, she's doing a DNA extraction. So she's using that kit that we first saw in the lab tour, and she's using the important tools there of the, as I said, the liquid transfer tools. So the pipettes, you would see these on TV. They always showcase these on CSI or Bones. And basically these are capable of transferring very small volumes of liquid. So one microliter, and to give you an example of what that amount might be, or what that looked like, it's almost the size of a pinhead, less than a pinhead actually, depending on what pin you've got. So Amara has weighed out the fecal samples that you saw in that Biosafety Hood, and she's applied them she's mixed them with a buffer. She will now go through a series of 4 steps where the first step is to break the cells open, so we release the DNA. Then once the DNA is released, we remove proteins and other components that are in the poop that are going to inhibit those downstream DNA tests that we need to do. And then the next step would be to go through a number of washes, and then finally you take I might just grab, Amara, can you hold maybe one of the green tubes up to or just pass me one of the green tubes? So, that's not, yeah. So basically, this is live. In the tube, there's a small membrane. And so the DNA, this isn't the right tube, but it gives you the idea. There's a small membrane and the DNA binds to there. So at the very end, we need to get the DNA off the little membrane inside the tube, and we use a buffer there. So once we end up with the DNA, we can go into the downstream molecular tests.
Okay, so there's a question about the PCR process.
Yep.
A little bit sciencey, but just to to ask you, because I'm not sure if you've already answered it. I got a little bit lost. How do you identify the region where the bacteria of interest is present? I understand primers can be used to identify known bacteria, but what about bacteria that we may not have sequenced a genome for? And so this is a question about, I guess, unknown things that could, that might cause harm.
Yeah, okay. And who's our fantastic scientist out there, or someone with a lot of knowledge?
Yes,
okay. Okay,
yes.
So Lisa, you're going to have to give me this in parts. Yes, when we do PCR, we can develop what are called primers, and those primers target a region or a piece of the DNA or gene of interest that we know will tell us that that parasite is present or not, okay? So that's looking for things that are known. There's ways to look for unknown organisms, and basically that gets into whole genome sequencing. It's not techniques that we do, well, we do them with bacteria, but not parasites. But you can just get the community of DNA, and then you can just sequence all the DNA or the RNA that's in the sample. And then you go through and search databases and see what they are closely related to. So with those instances, we don't know. We can find, say, new pieces of DNA that would group with microorganisms that we may have know before or know of, but we can never actually identify a new organism based on its DNA alone. Right?
Okay.
So a lot of
you know something about the jigsaw first before saying, oh, there's a piece of the jigsaw. You need to know that it's out there first. There's no way of just going, this particular sequence of DNA is an organism.
No, yeah. So you can't. You need to compare it to something that you already know about, and
right.
so that's what they, that with a lot of this is kind of a lot of viruses as an example. So you can take. I might just move away from the parasites that we work on, but you can take a sample, amplify, extract the RNA, which is a component of related to DNA. Yeah. And then you can sequence all the RNA that's in there. You don't need any primers. You just sequence everything that's in that sample. Yeah. And then you can take that sequence that you've got and compare it to all the known sequences from known viruses. And then it's like a jigsaw puzzle, and you match and group things. And then you can say, well, it matches it by 90%, so it belongs in that group. And similarly for bacteria, with the bacterial community and other organisms, we can approach kind of pathogen discovery, I guess you could call that. Yeah. But that's not an area of research that we work on. You're
a parasitologist. Actually, I've got a virologist who's just come on, and they have said that they know that virologists know that bats have a different immune system that allows them to maintain a viral load very differently to other mammals. Is this seen with parasite load as well? So is that question asking, can some species of animals take more parasite load than others?
Okay. Look, so that's an absolutely fantastic question, and actually one of our research questions. So yes, so there's evidence that suggests that bats have a really unique immune system, and the components of that immune system actually drive the viral diversity that bats have. And also protect them from the harm that viruses cause, that we see them causing in other hosts. So the immune system works and challenges all types of disease agents or pathogens, so parasites, bacteria, and viruses. And so one of our big research questions really is, this immune system, it's not all about viruses. There are other organisms out there. So we're actually looking at diverse parasites in bats, and so in the gut, in their blood, parasites that are related to the organism or the parasite that causes malaria. And then we're also looking at viral diversity in the microbiome. And instead of thinking about one disease agent and bats, we're trying to think about how everything works together and connects. And that's like our big research question, but we're chipping it away step by step. And our early research suggests that yes, bats have very diverse protozoan parasites, so parasites that cause malaria, Cryptosporidium, Giardia. We're seeing those similar trends relative to the viral diversity. How that links to the immune system is a whole other question that we're looking into as well, but it's a big, yeah, it's a career to address those questions.
So we come back in 20 years' time and you have the
Yeah,
answer.
come back in 20 years' time.
We're
I'm not going
looking forward
to
to.
about the state of universities at the moment relative
Well, we're looking forward to seeing like I guess the small steps that you make along the way that
Yeah.
all build up to the bigger picture, and
Yeah.
that's really, I guess, the lifespan of, you know, the career of a scientist. It's like you're answering a question, and it's and will it have an answer? But all the little steps that you have in the way all build towards that.
Yeah, and our question, yeah, and our outputs and publications leads to the next group of scientists who find and have different questions, and you know, what we find flags an idea in someone else, and vice, you know, like people who have found things before me have flagged ideas in us. So it's kind of like a continuing science discovery and research.
Exactly. The process is ongoing. Okay, sorry, there's some great questions. Thank you to our viewers. Yeah, keep going. Very excited. So are we doing the DNA extraction, or have we just done it?
I think we've just, yeah, it's going to go on.
Okay.
Are there any more questions?
Not at the moment.
No? Okay. I mean, we can flow back, but I don't know, what time were we meant to end, Lisa?
Oh, yes. It's okay. We've got a little bit more. We can go over a little bit, but have you got the GoPro set up on the?
Yeah. Look, if people are still watching, we can just swing back to the GoPro and my webcam.
Let's have a look at it.
We can, Amara stepping through. Oh, she wants something. No, she's got. Which one? Oh, 1,000. I have to give her one of these. See,
what's that thing in the corner there? Is that a centrifuge or?
Which corner? Oh, just here, just here.
Yeah.
Yes, it's a small centrifuge. So we've got lots of different centrifuges in the lab that they run at the same speeds. This one holds 12 samples, whereas the big one you saw early on holds 24 samples.
Mm.
So just our capacity for processing different samples. Yeah.
And so at the moment, you've got the bat poo is what you're trying to extract the DNA from.
Yes.
And are you looking for anything in particular in that poo?
Yeah, so not just the bat poo. So one area of our research is looking at parasites. So I'm particularly interested in gut parasites, and the two parasites I am very fond of are Cryptosporidium and Giardia. And so
I've got experience with those. I've had Giardia, and my daughter's had Cryptosporidium. So we're also, you know, on your team too.
Yeah. Okay. Cool. Great.
There is a question here. It's just come in. Do you have a list of parasites commonly found in bats?
A list of parasites commonly found in bats. Do I have a list in my head?
Yeah.
Do you know, one of the reasons why I started doing this work was because we had very little understanding of the diversity of parasites and reports of parasites in bats. And a few years ago, there were only like 6 papers in Australian species. So there's only like 6 papers that have been published on parasites in Australian bats. And so some of them include roundworms. Some of the work we've done, we found Cryptosporidium. They have their own species of Cryptosporidium, but they also carry one that can infect a number of different hosts. And they also, we've published the work on Cryptosporidium, and we've also published some work on, as I said, blood parasites, so called hepatocystis, that are related to the malarial parasite. That's by our collaborators. So I mentioned that our work spreads far and wide, and we work with lots of different people. And that work was done with Julianne Scher, who is in Berlin. And just to give a plug to the society and show how research works, the Australian Society for Parasitology really got that work going by supporting small grants and lab visits.
That's right. I remember that trip. We've got another question. Michelle, are parasites getting worse in volume and ferocity?
Yeah, no.
They've always been just as bad and just as prominent.
I know a lot of people think parasites are bad, but they're an extremely important part of the ecosystem. Mm-hmm. In some areas and in developing nations and in children, and where, you know, people don't have the medications or clean water, parasites are highly detrimental to communities. In the Western world, slightly different. To people, not much of an issue. Well, some areas, but different communities, but different people, different areas, less of an issue, at least in developing nations, and access to medicine. Generally, no, I wouldn't say they're getting worse. There's a lot of information out there on parasites being driven to extinction because we're driving their wildlife hosts to extinction, or we're changing the ecosystem, which impacts all the organisms that live in it, both parasites and their hosts.
I've got another question. Given the unique way that bats are able to handle pathogens, I have to say I agree with that. That's the first time I've heard of how I have never thought about a bat as being so clever that they can manage all these bacterial, viral, parasite load, with their own immune system.
Yep.
And the question is, why do you think there's a lack of interest in bat research in Australia? Is it a lack of interest because people just aren't aware of how clever their immune system is, or a lack of interest because people know that but they're not interested because they're bats and not people? What's your opinion?
I'm not sure if it's I don't know if it's a lack of interest. It's when we do our research, we have to do grants, we have to apply for funding. That funding has to be successful.
Yeah, sure. Okay. So maybe there's the competitive process being that only a certain number of things will be funded, and not necessarily that the scientists aren't interested. Maybe there's a barrier with what's getting funded.
Yeah, but over the years, like a lot of effort has gone into understanding viral diseases in Australian bats because of the couple of viruses that do harm humans. And obviously a lot of wildlife health studies, albeit bats or other species that live in our cities, people are interested in those organisms that are zoonotic and that are going to, well, not people that are interested, but that's the things that get a little bit higher, I guess, focus, that they're going to impact public health in some way. That doesn't say that there's not people out there and you're not hearing about research that people who are looking at wildlife health and asking questions around the significance or importance there.
Yeah, I think that actually the ASP have a good number of wildlife parasitology researchers. So hopefully that's an area that's going to be strong in the future and currently, under the spotlight. And there's a comment that says more funding is needed across the sciences in general. That's absolutely true.
Yes,
and also another comment which is in support of parasitology research, I think every mammal that goes extinct, which is a terrible thing, will take its endemic parasites with it.
Yes, so, indeed. Yeah. And that's what I meant by there's a lot of it. It's becoming more on scientists and parasitologists' radar that they're, we need to preserve parasites as well as their hosts.
Conservation is important.
Actually, so just coming just on that, the Tasmanian Devil program has a inclusion there to actually manage and preserve or conserve Tasmanian devil parasites as part of the management plan and conservation plan for Tassie devil. So it's and I think it's one of the first wildlife management plans that has considered that aspect, which is great. So great that Australia is leading the way again in wildlife parasitology.
Wow, that's great. So can we come back to you, Michelle, and I'll just see if there's any
physically back to me.
Yes, let's see your lovely face. And we'll see, are there any other comments? Just please write them down now. Otherwise, I think we probably need to, we've gone over time. It was such a fascinating live from the lab cross, and really live from the lab because we were right there and you were extracting DNA before.
Is it too late for people to join us tomorrow?
No, oh, for goodness sake, no. Yes, Michelle, explain what's going on tomorrow.
So if you're free tomorrow, Lisa will confirm the time because I can't remember, but we will be doing live from my lounge room. Not that you're going to see my lounge room, but we will be coming live from my lounge room and focusing on another project we have going, a citizen science project called Scooper Poop. And that'll be a little bit more of a workshop. So I think we've got some families and people signed up for that one.
Yeah, we've got a few people signed up for that one.
And talking about how you can get on board.
That's at 1:30
p.m.
Australian Eastern Standard Time. So that is 11:30 in Perth and 1 o'clock in Adelaide and Darwin. And all of these events are recorded on our Facebook Live. So if you miss them, or if somebody misses them and you want to share them, please go back to the ASP Facebook page and have a look through all of the videos.
And one more, slightly, yes. I know it's not an Australian Society for Parasitology plug.
Oh, another National Science Week.
National Science Week event tomorrow night with Claire MacArthur from Sydney, which will be continuing the parasites in urban wildlife and focusing on possums. And that's particularly with, I can't remember their name, Bushland Heritage in the Australian Capital Territory. So join in from there as well.
I've also got just a question. Is your lab doing research on bat ectoparasites as well? Now, ectoparasites, for people who don't know, are those who live on the outside of the body, not the inside.
Mm-hmm. Yeah, so we had some bat flies from a collaborator and up in Cairns actually, and they have been, the DNA for those is being examined by one of our collaborators, Julianne Scher, who I mentioned. Yeah.
So there are people looking at ectoparasites of bats. Yeah. Now, just before you go, Michelle, today we've got some more events for parasites online. We've got an art and science workshop with researchers from Charles Sturt University. They're veterinary parasitologists, and that's at 1:30 p.m. Australian Eastern Standard Time. And later today at 4:30 p.m. Sarah Preston is going to show us some parasite games. And then tonight at 7 p.m. we've got the La Trobe University researchers who will be talking about their work. And one of their parasites is a deer parasite. So interesting. So veterinary, and they also study malaria parasites there. So back on to you. Have you got any there's no final questions, but there's lots of thank you. So I'm also saying thank you. That was an awesome live event, and we will see you tomorrow at 1:30 p.m. for the workshop, 1:30 p.m. Australian Eastern Standard Time on Thursday. Is that right? Yes, Thursday the 20th.
Yeah.
Okay.
Yeah.
All right. And thank you so much, Michelle.
Thanks, everyone.
See everybody hopefully in a couple of hours. I'll see everyone. Bye.
Bye.
Okay.